What's up, party people? My name is Sunny Ray. This is the Sunny Ray Show. Welcome back. I've got yet another awesome, amazing guest today to share his story. His name is Ilya. He's the founder and CEO of a company called General Biophysics. The title is pretty crazy, right? The man who woke a frozen lobster. General Biophysics is a Framingham, Massachusetts biotech developing xenon gas as a therapeutic. Xenon, a noble gas long used in imaging, lighting, and anesthesia, crosses the blood-brain barrier and appears to shift the brain's immune cells. Microglia, no, I've never seen that word, into a state that helps clear. We're going to bring him on. Ilya, I'm not going to try and say these words, but tell us what you do. And maybe in short, like why should people care? What is general physics? And how on earth did you wake up a frozen lobster?
That's a lot to unroll. Hi, Sunny.
Yeah.
Thank you for having me. Yeah, our story started with the lobster because we tried to figure out how to make the seafood not to smell fishy when you saw it from the frozen state. And definitely we looked at the different cryopreservation technologies right away. So the issue with the cryopreservation that you have a lot of yucky stuff that you need to introduce as a cryopreservative, and then you need to take it out, wash it out somehow if you want to consume those cells, right, or bring them to the vital state. And we came across this article about using xenon gas as a potential cryopreservation agent. It's actually a very interesting structure. So the gas itself under pressure and with cold temperatures, let's say around 0 degrees C, can create structures with water which looks like flakes of snow. And it's not the big ice crystals, so they do not destroy cells. Also, there are potentially no issues with osmotic pressures, etc. This is what cryopreservative do in different biological cells and tissues. And we started to explore xenon. So xenon as a gas, it's in and out. It's a noble gas. It's in the headlamps, if you probably know, and other things, electronics. And now people trying to use it for the propulsion engines because it's very heavy gas in the satellites. So nothing related to biology. And we started to experiment with that gas, creating these structures with water. So we took the lobster and the shrimp and put it in a chamber with this gas and under pressure and in the cold temperatures, and managed to freeze lobster, or actually the small crayfish from Australia, which wouldn't survive winters and cannot be easily preserved. So what we figured out that we can actually then in 24 hours thaw it and bring back to life. So the lobster crawled for a while, lived with us for another 3 days, I guess. And it was filmed, and we thought, oh, we now can freeze humans, and sky is the limit. Unfortunately, it doesn't work that way in the complex organisms and tissues and cellular cultures. So this process is not very controlled, but we were along the way, and we try to preserve then biological cells and tissues using xenon gas. So this is where we developed this technology for preservation of human platelets for transfusion and red blood cells as well. So you can use xenon gas as a preservation agent. So you make cells numb to external stimuli. And because of that, the cells can be stored longer and safer. So this is how our story started.
Fascinating. That's fascinating. And then how did General Biophysics move from a food and blood preservation company into a neurological therapeutics? Like what was the actual kind of like turning point?
Yeah, that's actually two reasons brought to that. So the first one is that, yeah, we were, as any small company, we're looking for the funding, and we figured out that we are dealing with a logistical technology, which is not therapeutic, and we need to move ourselves towards therapeutic applications in order to be able to secure future funding for the research. So that's one very strong motivation. Another is that we looked at what we learned in terms of the biological mechanisms, and figure out that this way how we make cells non-reactive to signaling can potentially be used in inflammation management. And this is where also we started to explore the neuropreservation by the xenon gas, because xenon been approved in Europe around that time for anesthesia, and people reported that it has a neuroprotective effects. It's like it was even considered ideal anesthetic at that time. And we looked at neurodegeneration, so specifically Alzheimer's disease, and discovered during the research with animal models that we can modulate, as it's called, to change the phenotype of the immune cells in the brain. And that's all based on the mechanisms that we discovered with the blood cells and specifically platelets.
Wow. Okay. Can you tell us about like the moment I guess you went into that, but like about the lobster. But can you like what did that actually prove scientifically? Like the lobster story you kicked it us off with. Can you because you're saying that it's not maybe as transferable or as easy to bring to humans, but what did you learn from that experience that maybe in the future might be beneficial for longevity?
Yeah, so one of
Or
the things that definitely just a simple cryopreservation. Oh, we do not build the ice crystals when we freeze the things, and they wouldn't puncture cells, etc. But at the same time, we experience that with the xenon gas, how we create this clusters, these structures, is not very controllable process. That's why, as a cryopreservation, this technology wouldn't work fine for, let's say, preservation of the blood cells, because you just get a soup out of it. So you destroy all the cells, even if you're trying to use it in a liquid media. However, if you do not freeze stuff, and if you create a significant concentration of xenon gas around the biological cells, we discovered that you make these cells less susceptible to the external signaling. And we started to explore that mechanism, what's actually going on the biophysical mechanism. It's very interesting, because you can imagine the cellular membrane as an ocean with pieces of ice in it, which is called lipid rafts. And when these lipid rafts increase, there are a lot of signaling happening on their surface. So if you disrupt this formation of this lipid rafts and make the membrane of the cell much more fluid, then you can actually make these cells non-reactive to all the stresses that they experience or signaling from other cells. And this is a biophysical mechanism that we discovered at the time, and we thought, oh, it's interesting. We make cells non-reactive to this stimulus that would be inflammation cascade, for example. That's the one. And from this biophysical mechanism, we went to the exploration of the different models for Alzheimer's disease, mouse models. And actually, we completed the study and published in the beginning of the last year in Science Translational Medicine. That article is very scientific about the microglia and microglial phenotype. Microglia is immune cell of the brain. And the idea is that we can use xenon gas and high concentration of xenon gas that we can deliver to the brain. And at that time, we would be able to make these cells in a different format. So they do not create the inflammation, massive inflammation that would be damaging to the brain. And on the other hand, they're active enough to clean up the bad stuff that can be formed during the disease, like in the progression of the Alzheimer, like amyloid plaques and tau. And that's the mechanism we discovered for neuropreservation in brain in Alzheimer's disease. However, microglia is immune cell of the brain, so it plays a significant role in many other diseases like ALS or multiple sclerosis or others. And you can imagine that if you look from that standpoint, you can have a platform treatment technology that would have a multiple indications.
That's awesome. Really interesting. Was there a point where the platelet preservation business could have, become the whole company before, neuroscience pulled the focus?
No, I think it's still a very interesting approach, and there is a demand for preservation of platelets because platelets can be stored just for 5 days. Now the people extended it with a different pathogen and activation technology to 7 days, but still you cannot cool them down. So that's the problem of platelets. And if you cool them down, they got activated, you transfuse them to people in order to stop bleeding, and they immediately cleared out of the circulation in the bloodstream. So that's a problem of the platelet storage, how you can stabilize them or cool them down without activating them. And Xenon technology that we developed with the incubator and the bag system exactly does that. So you can make these cells in numb to external stimuli, that would be here cold, and you can store them for 14 days instead of 5. So the demand is big in the transfusion industry, and we're still trying to do that. Hopefully, with NIH support, we will be able to initiate clinical trials beginning of the next year.
And why build it that way instead of licensing the science out and, staying narrow?
Yeah, so it's a very interesting question. On the one hand, we are dealing with the repurposing of the drug. And when you repurpose, people ask, okay, what kind of composition of matter patents you have? We don't, right? Because xenon is a noble gas. On the other hand, you're looking at our patent portfolio. It's about ways how to make xenon treatment for therapeutic purposes or for storage economically and physically feasible, because it's expensive gas, so it would be very expensive to treat people if we do not recapture or reuse this gas. And also, it's a rare gas, so you do not have enough gas in the atmosphere in order to pursue all indications if you just waste it back, right? So in that regard, what we develop, we are working on identifying and specifying methods for treatment. We know that treatment of Alzheimer is a different story than it would be treatment of opioid use disorder and other indications that we are focusing on with the clinical program. And because of that, we are saying that, look, these are different protocols, and with different protocols, we create different pieces of IP, how we treat people as a method. And another part of the story is that this system that we develop, it doesn't require potential anesthesiologists to administer the treatment. It's inhalation machine that's completely automatic, and it also has ability to recover gas after the procedure, so we would be able to minimize the cost of the treatment. That's very important in order to make the business case work for this. And we know that there are a lot of companies try to explore xenon before for the different indications, including Air Liquide, who put this on the market in Europe for anesthesia, and it never caught up. And why it happened? Because of the lack of this recovery. And we figured that this is where our IP is valuable, and this is where we develop in the systems in order to bring it to the market, so people can say, oh, this is the business case behind it.
Interesting. And Nearly is largely on non-dilutive funding, right, through NIH grants. What did that path teach you that, maybe a venture-backed founder might never learn?
Yeah, that's very important. And I would say that we live in a unique country that allows to do these things for the new investigators. And you just need to be persistent enough and know the process and figure out that you can actually build something from scratch if you have an idea, and be able to get people interested around you. So very important to start with identifying the proper collaboration. So as a founder of the company, you have a very originally limited credibility, and you're trying to establish this credibility, scientific credibility, with working with academic institutions who already develop something in the areas of your interest. So you're looking for these people, and usually academic people are very open to this type of collaborations because you bring them new ideas, and you bring them ability to receive non-dilutive funding as well for their laboratories. And if the idea is cool, like, oh, xenon gas, like for treatment of such a difficult disease to treat, like Alzheimer, you finally would find the people who are might be interested and start working with you in order to do something like that. Then you need to generate some preliminary data. So ideally, if there are some publications available, but working with such collaborations, you can actually do some experiments, and these experiments demonstrate that you have something. And as soon as you do that, then you start applying and applying. So usually the process never works from the first attempt. I would say for Alzheimer, we applied maybe 5 times before we got the grant. And as soon as you are in the system, as soon as you have the good score on your phase 1, let's say STTR or SBR grant, then you can start from there, right? So you're doing your experiments, you developing new data, and very important to be very kind of accurate in terms of how you manage funds, how you deliver what you promise to deliver, because you develop your credibility with the agency. And as soon as that completed, then there is a phase 2, or there are a lot of other resources. We work with NIH, and they provide tremendous amount of resources that help entrepreneur, like I-Corps, for example. This is where you get the stipend and you work with your potential customers, and you're trying to build your business case. Or different grant programs, including the grant contracts, where you can actually build something really big besides what you can apply for as a small business administration grants, etc., etc. So this is how we started. And for particular technologies that wouldn't create the significant excitement with venture capitalists, because they operate within the quite specific business model, you can get your venture started, and you can generate even some early human data that you can use later on when you go around and looking for a dilutive investment.
And for someone who's only ever heard of xenon as, let's say, a gas in some light bulbs or whatever, how do you explain what it's actually doing inside the body?
So inside the body, it's the gas, noble gas. It gets through the lungs into the bloodstream. It's relatively not dilutive in water or water-based substances. That would be blood. However, it's very well dissolved in lipids. And that's actually brings this idea that for neurodegeneration, for the conditions in the brain, it might be very effective therapeutic, because it gets through the blood-brain barrier pretty quickly, and it's dissolved in the fatty lipids of the brain pretty easy. So you can build the concentration in the brain, and it's very, very quick. So you give the inhalation, and let's say 2-3 minutes later, the patient already has a concentration, therapeutic concentration, which is there. Very important that the xenon is a multi-target drug. So because of that, we can talk about like engaging the specific targets like an NMDA receptors, GSK3 beta, the microglial phenotype changes, etc. But it's also multi-target, so it's actually has this complex action, and because of that, it's relatively safe and effective for the different indications. You kind of not engage in just one target and then the whole body trying to compensate for that. You engage in the multiple targets, and you see how the entire disease progression actually can shift. And then as soon as you stop supply of the xenon, it gets out of the body. So it's this concentration drops very, very fast, also within maybe 5 minutes in the human body. The people stop feeling any kind of euphoria or the sedative effect of the xenon. Actually, not sedative, it's relaxation. And at that time, you can say that, look, we have some prolonged action, but the drug itself is not metabolized and it's not in the human body. That brings another point that it's very, very safe because it's in and out. You cannot say that, oh, there are some side effects because the residual concentration is still there and there is some metabolic changes that initiate those side effects. And then if you want to kind of build up on therapy, you just administer it repetitively. So if it's not the single inhalation, in our case for the complex diseases, you do that, let's say once a week, and you continue for a certain number of weeks in order to see the efficacy. This is what we established in animal models, and we really hope that the similar effects we will observe in human in the clinical trials.
And I was going to ask you also, so the gas recovery system means you're reusing xenon rather than venting it, right, after each session. So what does owning that piece like buy you that outsourcing would not?
Correct. So the important part here is that if we can build this as a vertically integrated venture, because if you have control over the, let's say, recycling of the gas, that's important for the business model as well. In the pharmaceutical industry, the simple model is that you invest a lot in development of the drug, and then when you manufacture this drug, let's say with the partners, you have a very significant margin on this drug because the production and manufacturing of the pill costs pennies, but you sell it for much more money. And this is where the business model for the pharmaceutical company is. In our case, we are closer to the medical device company, so our margins are smaller because the xenon is expensive gas by itself. And by actually recycling gas, putting and doing it in-house, we can issue the next batch of the gas at a much lower cost for the company. Because of that, this whole thing starts making sense. We are not only selling the inhalation machines, but we also selling the drug, the xenon, as a consumable. And if we can do that at the reasonable prices for the therapeutic agent and for the patients, but also would be able to minimize cost internally. That's a significant part of the business model. So this is our strategic thinking that we would need to, first of all, manufacture and produce and supply and service the inhalational systems, but also we would be able to recover the gas and offer it to our customers when the cost of this gas for ourselves is much lower than we can buy from the market.
Yeah, that totally makes sense. Interesting. And then you've also talked about, the tension, right, between like staying narrow and kind of building a wider platform. Where have you landed on that question, Ilya?
Yeah, that's a very kind of interesting question, and it always a little bit going back and forth. Should we focus on the one indication and just bring it to the market, or we need still to pursue the research for other indications and have a platform technology? And in our case, it's a combination of both. So we're definitely picking up the indication for the clinical development when we can, first of all, get some funding and support for that, and also bring it to the market faster. And when we are approved for one indication, we can potentially allow off-label use and explore other indications for other diseases, generating the additional clinical data. And then when we actually explore this platform for other indications. So this is our strategy at this point. And I mentioned that we are working on opioid use disorder indication for xenon gas inhalation. So this is what we're doing because it's actually easier to demonstrate efficacy for this indication than for Alzheimer, where the variability between patients is very large, and you need to have a very significant amount of the clinical research in order to demonstrate efficacy and get FDA approved. So as soon as we go through this first indication, opioid use disorder, we can actually use this as a platform, so we can expand another indication, allow doctors to administrate or try to use it with the patients who are have different conditions, but we have a significant data demonstrating that efficacy probably there. On the other hand, when you work with government funding and writing grants, you would like to explore different things in parallel, because it's still competitive programs. And because of that, we are exploring that with different indications. We can apply for Alzheimer programs and doing some clinical research in Alzheimer area or neurodegenerative area, as well as we are working with models and now moving into the clinical stages for opioid use disorders. This is just one example how users use this platform in order to be able to survive and move all things in parallel.
That makes a lot of sense. Ilya, just to like help me land the plane here, you've been a great guest. Thank you all for tuning in. But I have one last question. So if a clinician or like a scientific entrepreneur is watching this and wants to do something like what you've done, right? Which is start a company from a real kind of like unexpected discovery, what's the first thing you'd tell them?
So the first thing I would tell that definitely in my case and in the scientific entrepreneurship, you're looking first at the technology, and then you're trying to find the proper application for this technology. I know that if you start, let's say, the restaurant, you're looking completely different way, or if you working with the consumer goods, then you would be starting with the market demand, etc. But with a scientific entrepreneurship, you always start with the technology, and then you're trying to find a good application for that. This is how it works, because first of all, we're scientists, and then we start to be the marketers. So this is where when you look at the technology, try to consider what it does in terms of its functions. Let's say, try to put it in pieces and say like, oh, okay, so what are kind of functions I can use in order to explain what's going on. If it's a biological systems, what kind of functionality and biological mechanisms can be explained with? Or if it's a technical system, you're looking at different parts of the system and trying to figure out what kind of functions performed in this system. That gives you the notion that, okay, now the function can be generalized. I can find the applications for this function because this is not already your specific thing. We started with Lobster. So the function is to preserve biological cell. The function is where this function would be necessary, preservation of the biological cells more than anywhere else. And it is blood component preservation, because transfusion industry is a major consumer of such technology. So this is how you start. And as soon as you know where your pilot application is or champion application is, then go and find the collaborators, because with the collaborators you can do a lot, and it gives you the right credibility to develop in this area. And as soon as you do that, ask for government funding. There are a lot of resources, again, in the US, I believe in other countries as well, but in the US, it's a huge program with NIH and SF where you can actually get in. It's very objective evaluation, so you need to be persistent. You learn when you apply, and again, it took us like 5 times to apply before we finally got to the winning proposal. And this is where you develop your capabilities, and you can start the company right there.
Ilya, awesome, man. Thank you so much for your time. This has been super interesting. Thank you all for tuning in. Ilya, hang out for 1 minute. I'm going to bring the stream to an end. Yeah, I've got a few more interviews coming up later today, so stay tuned. Bye-bye, everyone.
Absolutely. Thank you.